Hi, everybody. Welcome to our next mini lesson in respiratory system physiology. These slides are going to cover the external and internal respiration processes of gas exchange at the capillary beds in the lungs and the capillary beds in the tissues. So again, here's this kind of overview navigator picture. So we just got done, hopefully got done covering the pulmonary ventilation lecture. If you haven't done that yet, maybe stop this one and go back and take a look at that mini lecture that's talking about how the changing volumes in our thoracic cavity drive the movement of air into and out of our lungs. It's basically breathing, pulmonary ventilation. So now what we're going to do is we're going to talk about this gas exchange that's happening here in those capillary beds of the lungs and out here in the capillary beds of the tissues. So this is what we call external and internal respiration. So external is the gas exchange with the air that's external, right? So we're breathing in external air into our lungs. We're doing gas exchange between our blood and that inhaled air. And then the gases get picked up by our blood and that goes out, from our lungs back to our heart, out to our body. And that is our internal respiration, is the gas exchange from our blood to your interstitial fluid, which will go to all of your cells. So in this lesson, we have a couple more old dead guy physics laws we have to take a look at. We're going to take a look at Dalton's law of partial pressure and Henry's law of gas solubility. So we are going to take a look at Dalton's law first. So if you saw in the pulmonary ventilation mini lecture video, I used the 760 millimeters of mercury as the pressure for the atmosphere. So atmospheric pressure, we saw that changed a little bit inside of our lungs, the intrapleural pressure when we were talking about 759 to 761, moving air into and out of the lungs. So when we take a look at our atmosphere, it is a mixed gas. It is not 100% pure anything of one thing. Its major components, and we can see them listed in this table, are nitrogen, oxygen, and a lot of other stuff. So typically you'll see percentages, maybe like 80% nitrogen, 20% oxygen. So that adds to about 100, but it's not quite exactly that. So in this top column here, this inhaled dry air, we can see it's about 78.6% nitrogen, about 21% oxygen, and then we have even way smaller percentages of carbon dioxide and water vapor. There's some other gases out there like argon and other things that aren't really relevant to our respiratory system. So we're mainly just going to be concerned with carbon dioxide and oxygen. And so one of the major reasons why it's carbon dioxide and oxygen that we're concerned with is because, one, they're soluble, lipid soluble, so they can move across the membranes, right, through those phospholipid bilayers. So that's one of their characteristics. Plus, they are also soluble in water, in the watery components of our blood. And nitrogen isn't so much. It's not very soluble. It doesn't go into solution as easily as carbon dioxide and water. And then water vapor is just a very small percentage of the gases we're going to be looking at. So we're primarily concerned with oxygen and carbon dioxide, but we have to kind of take a look at the bigger picture. And so that's why we're talking about some of these other gases. So when we take a look at whole atmospheric pressure, right, 760 millimeters of mercury. Remember when we were talking about pressure in Boyle's law, pressure is the total amount of times that all of the gas molecules are ramming around and bumping into the container. Our atmosphere is like a container, right? So we have our atmospheric gases. And that 760 millimeters of mercury pressure is calculated by adding up all of the pressures of the individual gases that's part of our mixed gas atmosphere. So Dalton's law of partial pressure says if you were to add up all of the pressures of the individual gases, that should total your total pressure of the container, in this case, our atmosphere. So if we were to add up, right, so 597, so 597 for the partial pressure of nitrogen, plus how much of that pressure is being contributed by oxygen? 159, plus How much is being contributed by carbon dioxide? 0.3 plus 3.7 for water. And if you did your math, beep, boop, bop, boop, 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 we would get 760 millimeters of mercury. And you can do that across the board for all of these in this table here. So what that tells you is of the 760 millimeters of mercury partial pressure, 597 of those 760 are due to the nitrogen molecules bouncing around. 159 is due to the oxygen, 0.3 is due to carbon dioxide, and 3.7 is due to water. Now there's some other gases they didn't put in there, but that's kind of the idea. So if you have a mixed gas, the total pressure of that container of mixed gas would be the sum if you added up all the partial pressures. So for example, if our atmosphere was 100% oxygen, 760 millimeters of mercury, if it was just pure oxygen, all 760 millimeters of mercury would be the cause or the source of that pressure. But that's not the case. That would be really bad for our atmosphere if it was actually that way. So we can take our mixed gas atmosphere, about 80% nitrogen, about 20% oxygen, and less than 1% of the other things, and then we just kind of divvy that up for who is contributing what to the partial pressure. Now, in the terms of applying this to respiratory physiology, these partial pressures, the PCO2, the PO2, which are the two gases we're primarily going to be concerned with, you can think of these partial pressures as almost like their concentration. How much of those gases are in that particular mixed gas? So in this top row here, we can see the inhaled air, which is just our dry atmospheric air, has oxygen about 159 units. It's not a percentage, so it's about 21%. We're going to be dealing with the raw numbers of the partial pressure, the millimeters of mercury. So it's about 159, and carbon dioxide is about 0.3. Now, when we take in, if we're to go in and take a sample of the air in the alveoli, how do those numbers change? So we can see, again, primarily just focus on the oxygen and the carbon dioxide. Our oxygen levels go down a little bit, which kind of makes sense because we're absorbing that oxygen into our blood. But take a look at the carbon dioxide. It goes up a lot, like a lot, going from 0.04% to 5.2%, or from a 0.3 partial pressure to a 40. That's a lot. And the reason why is because we are constantly dumping CO2 into our alveoli from our blood, and every exhale does not completely get rid of all of that air. And then we inhale again a few seconds later. So we're kind of rebreathing some of that carbon dioxide. Then we exhale, we get enough of it out, but then we take in another breath. And so we're kind of just hovering some CO2 there in our conducting zone and our respiratory zone. And so that's why the CO2 levels get so high, but they're not so high that physiology is not going to work. So these numbers are actually normal. And then if we take a look at the exhaled air, we can see the numbers, the CO2 and the oxygen change a little bit. So a little bit more oxygen, a little bit less CO2 if you were to measure that air that's coming out. So 100% of our atmospheric pressure is 760 millimeters, and we can see how we divvy that up. We can see what part of our mixed gas atmosphere contributes to that concentration. And I said, you know, we're mainly focusing on oxygen and carbon dioxide, but the bulk of the air that we breathe in and breathe out is nitrogen, right? Look, we're in the 70%, 74 to 78% of our inhaled and exhaled air, and the gases that are in alveoli is mostly nitrogen. But we don't, nitrogen is not a part, it's just inert. It's just kind of along for the ride. It comes in, it goes out, it doesn't really do anything under normal circumstances. Maybe we'll talk about some abnormal circumstances where nitrogen can actually become a problem. All right, so this is Dalton's law of partial pressure. The other law that we need to take a look at is called Henry's law. And Henry's law has to do with solubilities of the gases into a liquid environment, and how when you increase either physical pressure or partial pressure, that will increase the solubility of that gas. So we kind of have a couple diagrams here on the top. First two, it's like this generic plunger with some liquid, and then a real life example of a carbonated beverage. So let's say you were on the left hand side here. So in this right here, we have a container of fluid, we have some gas molecules, they're kind of at an equilibrium, and then the plunger goes from this position down to this position. So bring back in Boyle's law. If we're decreasing the volume, what's going to happen to the pressure in that container? The pressure is going to go up, right? It's the pressure on the gases. So when you increase the pressure of this container with this equilibrium between gas molecules in the air and gas molecules in the liquid, what you're going to start seeing is more of those gas molecules are going to be forced into solubility, trying to create an equilibrium. So if you'll notice, now that we are at equilibrium, we have the same amount of gas molecules coming into solution as we have gas molecules going into the atmosphere. And so it's going to be at equilibrium. So that's if we're to increase pressure. Decrease the volume, increase the pressure. So in a soda can, when you go to the canning facility, you take your sugar water, right? That's basically what soda is. It's just a whole bunch of sugar and some coloring and flavor, all that, whatever. And then if you want to make it carbonated, they put this sugar water under really high pressure with some gases. It's carbon dioxide is your carbonated beverages. So they're just cramming and cramming and cramming carbon dioxide under high pressure into individual cans. And so that's kind of what's happening in this A picture is we are increasing the pressure in this closed container, which is forcing the gases into the solution. All right, so then you have your sealed can because then they seal it. And so now you have all of these gases in solution in your gas container. I don't have carbonated beverages in my house, so I could show you an example, but if you have a carbonated beverage, either a can or a bottle, you know the bottles work really good. If you have a brand new bottle of something that's carbonated, you'll see there's no bubbles. It's not fizzy like it is when you open it. But then when you open it, you break that seal. The instant you break that seal, you see all the bubbles start to show up. Well, why is that? Well, when you break that seal on your bottle or open your can, you've just basically released or opened up the volume of that container to the volume of our entire atmosphere, right? You've completely taken the lid off. So that's what we're seeing down here is the volume has gotten huge. We've opened up the can to the atmosphere, which means the pressure on these gas molecules are going to decrease. So just the reverse of that part A, as the pressure decreases, the gas molecules are going to try to create an equilibrium. So we're going to see a lot more gases leave, a lot more of these dissolved gases leave out of solution to eventually try to make equilibrium with the atmosphere. Now, CO2 is a really, really small, it was like 0.03% of the atmosphere. So when you crack that can or undo the lid, all of the CO2 in the soda is trying to create an equilibrium in the atmosphere. Now think about what happens when you leave your bottle of soda or cup of soda or a carbonated beverage out for long periods of time, like the next day you go to drink it, it's not carbonated anymore. And the reason why is because all of the little gas molecules are leaving out of your open canister, can, bottle, whatever the container, to try to create an equilibrium with the environment because the volume just got as big as the earth, right? So that's Henry's law. So Henry's law, again, just to review, is dealing with the effects of changing pressure, usually by a volume change, right, because of Boyle's law. So changing pressure will impact the solubility of that gas. So if we increase pressure, that will increase the solubility. Okay. If we decrease pressure, that will decrease the solubility, right? So now what we're seeing in this example is the physical pressure of the volume changes and stuff, but this also applies to partial pressures. So if we were to increase the partial pressure or say like the concentration of a gas, that would increase the solubility. Okay. Also along with this idea is how soluble is the gas in the first place to a liquid environment. As I mentioned before, oxygen and carbon dioxide are really soluble. Carbon dioxide is the most soluble out of those three major gases. Oxygen a little bit less, but it still has some solubility. But nitrogen isn't soluble really at all. That's that one gas that kind of goes in and goes out and doesn't really have any role to play in our respiratory physiology because nitrogen is not very soluble at all. in liquid environments. But carbon dioxide is a lot and oxygen is a little bit less than that, but it's soluble enough so that it can go into our bloodstream. All right, so that is Dalton's law of partial pressures and Henry's law of solubility of gases. So both of those ideas are going to work together to describe and help you understand how gases move from our gaseous alveoli into our liquid blood. All right, so this diagram, this is from an older 2009, so it's quite an older book, but I think the diagram you should have in your new editions should be similar enough. So what we're going to do first is I'm going to set the stage for what the environment is of your inhaled air in the alveoli and your incoming blood from your pulmonary circuit coming from the heart. All right, so you got to remember, so we're coming from our body to our heart. So this is going to be our deoxygen. No, that's not a good color. I'm trying to find good colors for you guys, and I'm not super satisfied. Let's pick. See if this will go. So this deoxygenated blood, right, coming superior vena cava, inferior vena cava, coming from our body into our right side of our heart. We pump that out via our pulmonary arteries out to our lungs. And so this is the blood that we are talking about here. So this is the deoxygenated blood that just came from the body, right side of the heart, right ventricle, pulmonary trunk, out to our pulmonary arteries, to our pulmonary capillaries. So when we take a look at the characteristic of the partial pressures of oxygen, carbon dioxide, we can see, let's see if I can highlight, we can see 40 and 45. So what that means is the partial pressure of oxygen in this deoxygenated blood, right, that's going from our heart out to our lungs, coming into our lungs, is 40 millimeters of mercury. It's not a percent, so don't try to add things up to 100 because it's not going to work. So 40 partial pressure of oxygen, and CO2 is 45. Okay? Then when it leaves, so let's say the blood is traveling right next to the alveoli, when it leaves, let's see how that changes. So if we take a look at the PO2, it's 100, and the carbon dioxide is down to 40. Okay? So let's take a look at the changes that we see. So the incoming blood of PCO2 is at 45. And as it's leaving the pulmonary capillaries, getting ready to go back to the heart, to the left side of the heart, it leaves at a partial pressure of 40. And then the blood coming in, the PO2, comes in at 40 and leaves at 100. Okay, so we have a difference here. So if we take a look at just at the carbon dioxide, it comes in right next to the alveoli of having a partial pressure of 45 and it leaves as 40. So that means something happened to 5 partial pressures of the CO2. And if we take a look, it leaves and goes out to the alveoli. And then if we take a look at the oxygen, it goes in and we are gaining a lot of oxygen in this partial pressure. Okay, so let's take a look at the environment in the alveoli. And these numbers again came from that table on Dalton's law page they had highlighted, that we have oxygen as at 100 partial pressure and carbon dioxide is at 40. Now, if you'll notice, these numbers are very similar to each other. So if we were to, I'll do oxygen is normally red, right? So if we take a look at the oxygen levels in the alveoli, right here, as that deoxygenated blood is coming next to it in the capillary, we have this gradient of 100 over here and 40 over here. We go from high to low, high partial pressure of oxygen to low partial pressure of oxygen, and it will continue to load oxygen into the blood until it's reached an equilibrium. So when we inhale and we have oxygen in our alveoli, it will continue to try to load into the blood until it reaches an equilibrium. So that's why the blood leaving the pulmonary veins is at 100 millimeters of mercury, because it's matching what's in the alveoli. So then if we take a look at the carbon dioxide, Let's make that be blue. Carbon dioxide is a higher concentration here and a lower concentration here. So we have 45 in the capillary goes to 40 in the alveoli. So we are diffusing, things are leaving the blood capillary and going into the alveoli until it reaches an equilibrium. So that's where these numbers come from, of what are the saturations, if you will, of oxygen and carbon dioxide in the blood that's leaving the alveoli. It matches the saturations and the concentrations of blood in air, these gases in the alveoli. So I always thought that was super cool. So you have, whenever you inhale, all that oxygen in your alveoli will load up into your capillary until it reaches an equilibrium of 100 millimeters of mercury. And then as we are getting ready to exhale, the CO2 is dumping from the high concentration in the capillary to the lower concentration in the alveoli. It's diffusion. Things go from high to low concentration. They're going from high to low partial pressures. It's the same concept applies. All right, so that is, your pulmonary capillaries, and then the blood leaves your pulmonary veins, right? The capillaries, pulmonary veins, comes back to your heart. So that's this direction over here. And then it goes out into your systemic boink, right? So we're coming from your body out to your lungs. We just did that. From your lungs back to your heart, bringing in the highly oxygenated blood and going back out to the body. And now so everything we've just talked about, this is external respiration. That top part with all my squiggles all over it. So that is our concept of external respiration, the gas exchange that happens in the capillary beds. Now we're going to get down to the tissues, all different kinds of tissues. There's smooth muscle, some fat, looks like a neuron, maybe a cuboidal cell. It's just kind of a collection of tissues representing your body, your systemic circuit. So now we're going to come down and see what's going to take place here. So if we set the stage, what are the partial pressures of gases in our tissues? So oxygen is at 40, CO2 is at 45. In our incoming blood, oxygen is 95, carbon dioxide is 40. So here we have to set up our gradient. But before we do that, I'm just going to click to another slide real quick to say why do we go from 100 to 95? There is no gas exchange from coming from the lungs back to the heart, out to the body. It's in these big elastic arteries, pulmonary veins, aorta, all of your arteries. So why do we lose some of the partial pressure from this freshly oxygenated blood coming out of the alveolar capillaries to the systemic capillaries out in our body? I have a picture to help explain that, and then we'll get back to finishing up internal respiration. Here we go. So this is a close-up diagram of some of the anatomy stuff that we just got done talking about here in another lecture. So here is our terminal bronchial, right? So terminal bronchial comes in down here. It branches into our respiratory bronchioles. And again, we know they're respiratory bronchioles because they have little alveoli bubbling off of them. Anytime you have an alveolus, you know you're at the respiratory zone because you have gas exchange. But that's not what I want to call your attention to. What I want you to see are the systemic arteries. Let's see, get that highlighter back. That seems to work pretty good. So if we take a look right here, bronchial, ooh, that is not good. Let's get yellow. Black highlighter doesn't work. So bronchial artery, so this is part of the systemic circulation, right? So from the left heart via the thoracic artery. So this is blood that is feeding the tissues of your lungs. So it's feeding the connective tissue of your bronchial tubes. It's feeding the smooth muscle that's not shown here, but you'd have smooth muscle, blood vessels that need to feed the blood vessels, right? Some vasa recta. Now take a look at its capillary bed right here, right? So that's the capillary bed that's feeding that part of the tube, and look at where it's draining. Let's follow that. Oh my gosh, you guys, it's draining into the pulmonary vein. Deoxygenated blood from the capillary beds that feed the lung tissue is draining into that 100. So if all of this is coming out as 100 partial pressure, 100 partial pressure, 100 partial pressure, but we're having this drainage from your systemic circuit of your lung tissue. This is where by the time we leave the lungs, it's dropped down a little bit to 95 partial pressure because we're draining some of the circulation to the lung tissue itself. So here's another, if we're gonna, man, I cannot get away from that black highlighter. Yellow, there we go. So if we're gonna follow this, again, here we see another little capillary bed and draining into the pulmonary vein. So the reason why that's set up that way, it's just a kind of a shortcut. So why would we want to drain these systemic capillaries into a whole other vein to go back to the heart when we can just dump it right into this pulmonary vein that's right close by? So it's a little bit efficient design there. And we do drop down to 95, but that's still going to be a big enough a gradient for the oxygen to leave the blood to go into the tissue. So let's take a look at that. There we go. Okay, so now we're back to our, right? So this is our internal respiration. So again, here are those values that we just picked up, right? So our partial pressure of 100 goes to 95, and we just talked about why that is, the drainage of the systemic vessels and capillaries to the pulmonary vein. But our CO2 stays the same, so it's 40 leaving the pulmonary capillary, and it's 40 coming into the systemic capillary. So it's 40, 40 by the time you get to your tissues. Now, if we take a look at our tissues, we have the O2 of 40 and the carbon dioxide of 45. So if we take a look at those gradients, we can see. So if we are going from a 45 gradient in the tissues of the CO2 to a 40 gradient in the blood, there's our gradient. And so you're going to have an equilibrium of carbon dioxide going into the blood until, look at what it does, it's reaching equilibrium. So by the CO2 concentrations in our tissues higher than the CO2 concentrations in our capillaries, CO2 will leave our tissues and be picked up by the blood as it goes from this 40 coming into the tissues to the 45 leaving, because we have a higher concentration of CO2 in the tissues. Now, if we take a look at O2, right, we have a higher concentration in the blood, which is 95, and we had an oxygen of 40 in the tissues. So where's our concentration gradient? We want to go from oxygen going this way, and it will continue to cross this membrane, you know, the capillary exchange, right? Remember that from 232, until it reaches equilibrium. And if we take a look at that number, oh my goodness. If we take a look at that number, it's 40. So oxygen will leave the capillary and go into the tissues until it reaches an equilibrium with the tissues. And what's really cool about this whole thing is this is our systemic circuit, right? So now we're coming from all of our tissues back to our heart. And if we're to make a big arrow right to our heart and then back out to our lungs, look at those values. 40 and 45, 40 and 45. Isn't that so awesome? So this is what we call our gas exchange. So we have external respiration as a gas exchange at your lungs, where the oxygen is going from the alveoli, following its gradient into the capillary. CO2 is leaving the capillary, going into the alveoli, following its gradient. That oxygenated blood goes down back to the heart, out to all of your tissues, and then we get an internal respiration, where oxygen leaves the blood, goes into the tissues, following its gradient, and CO2 leaves the tissues and goes into the blood and follows its gradient, and then it does it all over again. So we have this continual, that's connecting to our cardiovascular system. Why do we have the pump and the blood vessels and the capillaries and all the exchange? It's because we need a way to deliver these gases. We have to deliver oxygen to our to all of our tissues, and we have to have a way to pick up that carbon dioxide. And then our lungs help us exhale that toxic gas that we do not want building up in our tissues. All right, so that wraps up our gas exchange mini lecture. So we have a couple more in this system. I'm going to talk about gas transport, what happens with oxygen and carbon dioxide as it gets into our blood. And then I will have a lecture on regulation, the neural regulation of your breathing and all that kind of stuff. All right, so I will see you next time.